All right, our second speaker is Sikiic, talking about de novo genome assembly using AI.
Hello, everyone. Thank you for to Chirag to invite me. So I really had very, very nice 3 days, and it's my first time in India, and I really like it. So today I'm going to talk to you about how we can use AI in genomics, and we started this approximately 5-6 years ago. And before I worked mostly with algorithms, and then I realized that I saw the increased interest of students in AI, and I decided, okay, it's time to switch. So de novo assembling is an old problem. It's a computationally difficult problem, but we are improving a lot in the last 25 years or more. So the idea is the following. So we want to read our whole DNA, but it is huge. The problem is that fragments of DNA that we can sequence are very short. So we need to find some way, algorithmically, to reconstruct the whole sequence. So we can imagine that you have one book. And what we can do, we would like to read the whole book. But we can read just a few paragraphs. And that's the problem of de novo assembling. So the approach is the following. Just copy, 40 books, and just put in some shredder or some kind of machines, or just throw a bomb. And you will see you have lots of fragments. And then, like detectors, you try to overlap these paragraphs from the book and try to reconstruct the whole human genome. So that was the challenge. And the first attempt, and it costed $3 billion. And they sequenced the human genome, and at that time, they resolved only 90% of the whole genome. But recently, a few years ago, Sergey Nurk and others, the parts of telomere-to-telomere consortium, they reconstruct the human genome. It was haploid 1, CAGM13, almost telomere-to-telomere, all chromosomes except one really nasty part, RDNAs. So this is still unresolved problems because there are many copies, they are very similar, and we don't know how many copies there are. But that gave us something new, so that we can read the human genomes, and then, as from Wikipedia, you can use AI. We can, because we have high-quality genomes, we can use AI. So how de novo assembly works. First, we have the complete genome. We cut it into small pieces, so we don't know the order of them. So we need to find, in the first step, some kind of overlaps. So using the information that the suffix on one sequence is equal to prefix to another one. Paul mentioned today the Bruin graphs. There is another approach I use is overlap layout consensus. So idea is to try to find the good overlaps, then construct the graph. We construct the graph in a way that nodes are reads and the overlaps are edges. And theoretically, if you can find the path to the graph, you can reconstruct the genome. So the biggest problem is how to find the real path. Theoretically, it is a Hamiltonian path, and it is NP-hard. It's not possible, so we use another approach. We try to make our job easier, try to simplify this graph. And when we can do this, we try to find some unique paths, and then these paths we call our assembly. Very often, it is fragmented because there are some regions that we cannot reconstruct. They are too complex. We cannot simplify and be confident that with some certainty that we can pass through them. All of today algorithms for de novo assembling uses some heuristics in most cases, and they try to pass through these regions, but when they have some certain confidence. So, and as you can see here in green, it's that part that we are looking for. Okay, so that's the classical approach. So, and this is one of assembly graph for chromosome 20. And chromosome 20 is actually one of the smallest ones and one which is not difficult. The difficult and nasty chromosomes for de novo assembly are so-called acrocentric chromosomes. So 13, 14, 15, 21, and 22, because they have these rDNAs, and they're very, very difficult. So we really need to find the path correct through this graph. So what is the current approach? The current approach is that you try to simplify this graph using heuristics. And then, even if you have some regions, you try to guess which part is better than others. And there are really some kind of magic how to tune the parameters of your algorithm that it works correctly. And I can openly say, nobody in the world can do this better than Heng Li. So I decided to try to use AI against Heng. So that was another reason we moved. So, you know, Heng can find the perfect parameters, but is it possible that AI can do something better than him? So that's the reason why we moved also to AI. So I got the idea why we were working on Raven. Raven was, it's the fastest de novo assembler for nanopore reads, especially if you don't worry about diploids. So just to have the fast and with not so many data reads. And while Robert and I worked on Raven, I found something that was interesting. So when we looked at our chromosomes, I found that you can see these red edges. So these red edges that actually connects, in this case, it's some kind of bacterial genome and with some plasmids. So we found that if we draw the graph and if we see these elongated edges, if we cut them, we will have a good solution. Okay? So it required two things. First, to notice something like that, and then think and construct the algorithm to solve this problem. But the question is, why I need to do both tasks? Why not to give the same task to AI? So we started with an approach that we tried to utilize, that now we can reconstruct some of genomes telomere to telomere. And it required a lot of manual curation. I will tell you a little bit later about some other approach and taking manual curation. So even for CAGM13, it was not achieved by an algorithm. At the end, it required a lot of manual curation. I was involved in manual curation of AG02 and some other genomes. It really takes time looking at IGV and trying to find and understand what is the real sequence. So, but when we have this sequence, what if we can find a way how we can train our algorithms or AI to find the paths in this graph? So what we can do, we can have a, for example, CAGM13. We can sample reads, and we can sample so many times as we want. We will have a different read set. Construct the graphs, and then we know the ground truth because we sampled from the real genome. And then we know the ground truth, and we can use AI. So the idea is to get unfragmented graph, and actually the idea was to find no edges that we can traverse that can help us to find the final path. So it required a lot of training, But once when you achieved this, the task is much easier. So what we did? So we used BDBIO. So we started with BDBIO because at that time hi-fi reads were the most accurate one, and we tried with BDBIO reads. Now I will start with nanopore reads because they are longer, and with correction you will have much smaller graphs. But we started with hi-fi. So it was important to have a good simulator. So we used the PBSim3. It is not perfect, but you need to accept some errors, and sometimes some noise in AI actually helps. So, we tried to achieve the same length distribution, everything, and we started to simulate reads. So what was the idea? So the first step is always overlap. And I really believe, but we are still not close to that overlap is all you need. So if we can perfectly overlap all reads, we don't need layout, and, but still we are far from that with repetitive regions and all kinds of problems. So we will construct graph. We used HiFSM because at that time it was the best tool to using, and then we will label the right parts using the reference, because we know what is the right path. How? Because if you have a read from one region and you see that you have a read that comes from the neighboring region, you know the overlap is correct. So that was the idea that actually helped us to build the whole story. Okay, but how we can use AI? Can we use transformers like in natural language processing or convolutional network, or how we can do in graphs? So we needed something that actually emerged slightly before we started with this effort. It was graph neural networks. So the idea of graph neural networks is the following. So in a graph, you have nodes, you have edges. And you want to learn something about your local structure, or maybe the whole graph structure. But how you can learn if you don't know actually how many neighbors you will have? And you don't know the order of these neighbors. So so this kind of algorithm should require two things. First, that it should be invariant on any kind of permutation, and you can have any kind of, any number of nodes. So today, the dominant paradigm is using so-called the message passing, and then to aggregate this. So you're using information from all of neighboring nodes and edges, and in some way you can aggregate. How you can aggregate that it keep permutation invariant? So you can find a minimum, maximum, you can sum them. So we used just maximum. And what we do next? So how we do this? So just collect information from all nodes around you, and you update your current state or your current embedding of the node. In a similar way, sharing information from nodes, you can update the representation of your edge. And you are doing in each step of graph neural network. So in each step, we are computing using some operation the new state of edge and node. So how the model look like? So we have some node features, we have some edge features, and then we do just linear projection, and then we have a graph neural networks. So for node features, it was easier. So you can use degree or you can use any other measure about nodes. For edges was difficult, so in the beginning we will just use some random information for node representation. So the GNNs can have multiple layers. Actually, it depends how big is your graph. So because in GNN, in the first layer, you are learning information from your first neighbors. You aggregate this information. In the second one, you aggregate information from the second neighbors because they send their message in the first round to you. So it requires to have some depth of these graph neural networks. And in the end, you have for each node and edge, you have some representation. So these are representation for nodes, these for edges. And in the end, you are trying to predict something. What we try to predict, we try to predict for each edge. Can we use this edge and achieve the perfect assembly? So we used gated GCNs because at that time they performed the best. The idea of gated GCNs is that you have this additional parameter here. So for example, for edge, if you want to update the current representation, you have the current representation, you have here ReLU, batch normalization or something. So you have metrics and you multiply all neighboring nodes, plus you add information related to edges. So this parameter you can see here, actually it's some kind of gate. It gives us information how much of information from this specific edge we can use. So some edges can have a larger value than others. And also we can have, we need to update edges, so we can do it in a way that you have the current edge and you have some matrices, so you multiply the current state and you can multiply the two nodes and which are connected with this edge. So this is the idea. You just add these other things. So you can see, and this is some kind of that you are adding the information inside. So this is the whole graph, and it was not easy to implement. It takes lots of time. One important thing, I will not go too much to technical details, is that we found that it is important to distinguish for each node incoming and outgoing edges. So in most graph neural networks approach before, people didn't care about this. But we found that it helps if we separate information when we calculate representation of this node using information from incoming and outgoing nodes. And you can see here the differences in blue and in purple. And for each of them, we have different matrices and we calculate separately all these gates. And here you can see the whole equation. So for training, once we established this, it was easy because we know the ground truth. So you can see here that you can have several ground truths. Actually, in the no assembling, I will show you later, most of overlaps are good enough. So, but there are few of them that you need to take care of. So majority of them are good, so you can have the several paths. For example, from this node, you can go to this one, to this, or you go to this. One important thing to mention, we didn't do anything to simplify the graph. So usually the first step is removal of transitive edges and some tips. We didn't, because to keep more information in the graph. And then, yes, we use the graph neural networks and some just last layer, fully connected layer, to try to predict value of edge, so it is valid or not, and we use just simple binary cross entropy loss, so it is correct or not correct. So here you can see the whole idea and presented in the paper. So this is just another and final trick. So in a graph, you have node, but you have reverse complement node, because you don't know how the machine sequencer read this read. So because it is like this, you have 5 prime, 3 prime, 5 prime, 3 prime, and you can rotate it in any way. You don't know. So while we are resolving graphs, we need to keep the both information. So actually we have 2 graphs that are going together. What we did, we use this information to add something to loss. We added this symmetry. Why? If I have a node and reverse complement, and I have another node and a reverse complement, the probability that I can go through this edge between them should be the same as the probability if I go a traverse in the opposite way. So we use that information to have these 2 binary cross entropy losses for node and reverse complement, but also try to put them to have the same values. This is just logits before the last layer. And decoding, it was easy because you just find some node with the highest probability and then go into direction, and we use the greedy approach. We tried beam search, but it didn't work so well, so we at the end kept with greedy approach. And I believe there is lots of way to improvement. So we worked only, because it's the first implementation, on haploid genomes. So as you can see here, we presented the percentage of N50 and NGA50 from the total. So the total will be 1. So we achieved from high on chromosome 13 better than high FSM. And we achieved the similar results on mouse, on the Arabidopsis, and some chicken. And it was interesting. And I think it's the still. If you see, the assemblies of human genomes are much better than others, because we trained on human genomes. But all people who develop algorithm train and create the hyperparameters using human genome. So the results are always biased to human genomes. So take into account when you So it was recently published in Genome Research. It was not easy when you come with something completely new to convince people, because people would like that you solve all kinds of problems, but this is not possible. And yeah, so there are some limitations. We found some ideas. I think this one is just important to show. So if you have the full graphs, and you can see that some kind of decision nodes, when you have some nodes which are not all, if all of edges are not the true ones, there is just 0.86% of them. But if you look the centromeres, it is almost 10%. So now we are focused on just the centromere regions, because other regions, the machine learning will learn without problems. So I really, I'm happy that I had a great team. So Lovro did most of work with help of Martin, who works now on deployed ones. We had the help from Xavier. Xavier was one of 3 first persons who introduced graph neural network and graph convolutional network. Kenji is the genius one. You talk with him and he gives you references and papers where you can find something. So it was really amazing experience working with them, and Thomas, who is a mathematician. All of them really helped us to achieve this. So before I finish, I started with the human genome project, and I would just like to present something different. So as a part of National Precision Medicine program in Singapore, when we try to sequence now 500,000 Singaporeans, we are developing some reference genome for 3 major ethnicities in Singapore, so Chinese, Malay, and Indian. And the first genome we decided to reconstruct telomere to telomere was the Indian one. So 10% population in Singapore are Indian descent, mostly from southern part of India. And so this is the first genome telomere to telomere of an Indian. So as you can see here, it's just NGA50. It's similar to other genomes. So you have, the first one is Chinese, the second one is our Indian, this is AG02, I think it was Ashkenazi Jew, and it was another Chinese, and CHM13 was Caucasian. And as you can see, the quality value of this one, it's above Q80. And at this moment, this is the most accurate reconstruction of the genome. And we are finishing, and I hope in 2 weeks we will publish this. So yeah, I have to just also mention JJ. Together we are working on this Indian genome, and these are all people who actually did job. And I really have to highlight Prasad, who did amazing job together with Josipa. Thank you. Yeah, I was slightly over time.
Questions? Maybe I'll start with one. So something that you mentioned, that when you're regenerating the read sets to train your network is essentially learning the human genome. Because have you thought about expanding the data so that you have multiple references?
Yes. So that was the idea we got after CHM13. Now you have more. You have monkey genomes and something like that. So with more data, we believe the method will be better and better. Yeah. And also, my actual idea is how we can expand this idea to some genomes that we have not tried to resolve so far. For example, I think someone asked about polyploid genomes, and my personal challenge are cancer genomes with aneuploidy and all of these. So instead of thinking about algorithms, I can always try to find some specific, I don't know, if some chromosome is Q part or something, I can simulate all of these and then try and give to network to learn.
Hey, Mile, nice talk. So the question I have is, do you have one large graph or do you have separate graphs for ambiguous areas?
So it is, so we create one graph, but sometimes if you are lucky enough, you will have some chromosomes already separated. Mm-hmm. So we start from one graph, but this is the output of the overlap. So sometimes it is clear, but in most cases, especially these acrocentric chromosomes or around centromeres, you have lots of edges and you need to resolve them.
Okay, so I think I saw on one slide you were doing partitioning using Metis.
Yes.
Will it make sense to, instead of that, will it make sense where we already have, through traditional algorithms, we know that this is how the assembly is going to look like, because those regions are not ambiguous. So if we cut there, then we'll make sure that the ambiguous regions are in one subgraph. And
yes. So that was one of the reasons we decided to move just to these centromere regions. Mm-hmm. So the graphs are much smaller. And on the other side, now we have GPU cards with more memory, so we don't need to use Metis anymore. So that was the reason I even didn't mention it, but it helped us in building. So Metis is a tool for partition of graph.
Okay. Thank
Probably
you.
the best one. Thank you.
Yeah, here. Very exciting talk. You started with the, correct me if I'm saying something wrong, you started with the human reference genome.
Yes.
You took your simulator generated reads and used that for training your neural network. So what if you had started from random sequences and for every training run essentially used different random sequences, simulated those reads and trained your network? Did you try that, or is that something that just would not work for some reason?
So, and it is just my maybe naive approach. So most of new organisms were a result of evolution. So they have many things in common. So it is not entropy that is maximum. So it's much better to start with known genomes because, for example, the differences between human and other mammalian genomes are not so huge. So I think it doesn't make sense to try with random.
Hi. Yeah, so my question is regarding variant detection. So does assembly-based approach works better than reference-based when it comes to detecting variants, like in highly polymorphic regions such as MHC region?
So, okay, maybe I am biased, but I think that assembly-based approach is always better, but we cannot afford it always. So, for example, for practical clinical, and someone mentioned, we still need short Illumina data because nanopore or PEG-bio are much more expensive, and you need, it's not only they're expensive, you need much more DNA. So especially if I really like ultra long reads, so you just need ultra long reads if you want to assemble anything now. But you really need a lot of material. So it is not only about price, it's about DNA, how much you need. So if you want to do this kind of assembly, you cannot use just tissue. You need to have a lot of them. You can produce some cell lines that you have enough data for this. So always, if you can do assembly-based approach, is better. But I think now the most practical approach is using some kind of pan-genome graph and trying to map reads to pan-genome graphs.
Hi, so I have a very naive question. So when you implement graph neural networks, how usually you come up with the initial graph which you build and then you...
Oh, so we use just overlap from HiFSM. So HiFSM outputs the one. So we did the same with our Raven algorithm, but it worked only on nanopore, and at that time the nanopore reads were much worse. So we decided just to talk with how you please provide us this output. So yeah, it was the reason why we decided to use HiFSM. So, but any kind of overlap and graph construction algorithm can be used in this case.
Thank you.
Okay. All right, let's thank Mila again.